Our next speaker today is Dr. Binh Pham, who will be speaking on behalf of Flagship 3.
Good afternoon, everyone. My name is Binh Pham, and I joined Sydney Vital in August this year. So it's my great pleasure to represent our fellows to talk about application of nanotechnology in the diagnosis and treatment of colorectal cancer. So with the nanotechnology, we aim at enhance the delivery of chemotherapy or radiation to the cancer site directly, and use the diagnostic with the nanotechnology to detect the lymph node metastasis, or being able to track the development of the and the staging of the colorectal cancer in situ during the colonoscopy. So this is a very busy slide, and this is just a snapshot of what our former research fellows have been able to obtain. Dr. Wei Dang, she applies a nanotechnology to design a liposome delivery platform with the addition of the low-dose X-ray to be able to release the chemotherapy to treat the colorectal cancer. And they already published in the practice journals of ACS Cancer Science and Nature Communications. More detail you can find in this publication. But if you look at the graph here, you can see that with the liposome they use in the cancer-carrying animals, they were able to inhibit the tumor growth significantly compared to the animal without liposome and low-dose X-ray. And another former research fellow, Dr. Yasser Gulami, he's also in the audience. He applies the nanotechnology to design a nano-radiotherapeutic for lymph node metastasis. And Yasser utilized existing FDA-approved inosine nanoparticles to be able to track the lymph nodes metastasis in the cancer-carrying animals. And if you can see here, they were able to detect the lymph nodes metastasis in the animal with the particles, while the particle were not observed in the healthy animal. So this will be able to help the clinician to be able to see the how much the cancer spread out. So since I started in August this year, we were in close discussion with Professor Alexander Engel and Professor Mark Malloy. We identified two main areas that close relevant to our clinical practice, that whether we can identify the adenoma or the staging of the adenoma during the colonoscopy. And as we know that what happened in the gut is very close in relationship with how it developed for the bowel cancer or bowel disease. So our target we set out is to be able to design a nanoparticles probes to be able to track longitudinal study in the in vivo study. From the previous study with Associate Professor Viva Howard and Dr. Emily Coben, using the nanoparticle in the metastatic ovarian cancer model, you can see here the particle highlighted all the metastatic tumor, why it's very difficult to see what's happening in the mesentery of the animal without the particles. And we try, as a proof of concept, we use fixed colon tissue from the colitis model. And you could see here from H&E staining, the particle stains a certain cell type inside the colon sample. And from Mark Malloy study with Macquarie Uni group, we were able to utilize the fresh intestine sample, and we treat the sample with 2 different types of the particles. They have the same core, which iron oxide particle, but they were labeled with 2 different fluorophore. One is rhodamine on your left-hand side, and one with near-infrared on the right-hand side. And you could see that the particle highlighted the colon, highlighted the small intestine differently with the 2 different fluorophore. So we are under process to study further with the HND staining and iron staining to look into the macroscopic what happened with the pathology where the particles stain. Then we will aim to translate this ex vivo model into the in vivo model using the IPC mean mice. So with the achievement we've obtained so far couldn't have been successfully achieved without the collaboration with multiple experts and multiple institutions throughout the flagships, and it's Sydney Uni, Macquarie Uni, UNSW, and Harvard University. And we are planning to engage the Olivia Newton-John Cancer Research Institute for the IPC mouse model. As you can see, the 2 fellows have achieved very successful research career with the support from Sydney Vital. They were able to achieve more fellowship and grant to continue their translational research, and the patent they lodged patent approval with Sydney Uni, and Wei Dang and her collaborator got the startup company with Canada. And with that, I'd like to acknowledge Sydney Vital for the research fellowship funding for me and for our former research fellows. And thank you for your listening.
Well, thank you, Ben, for a really interesting overview.